Good day, everyone. Welcome to today's Yellow webinar. I have a pleasure to introduce Laura Gragnani. She's now a tenure track researcher in the Department of Translational Research and New Technologies in Medicine and Surgery at the University of Pisa. Since early stages of her career, Dr. Gragnani focused her research on clinical and molecular aspects of hepatitis C virus infection. And I can say that her dedication to research resulted in a really impressive number of publications. In recent years, Dr. Gragnani uh, switched her research interest toward the notch signaling pathway and their role in cancer development and progression. Indeed, today she will present notch signaling in liver cancer. So please, Laura, the screen is yours. Thank you so much.
Yeah. Okay. Just a moment.
You can
Let me
share.
see. Yep.
Perfect. Okay, we can see.
Okay.
Can you? Okay, perfect. Thank you so much.
Okay. Thank you. Good afternoon. And first of all, thank you very much to Davis for his introduction and for inviting me today to give this seminar. I also would like to thank Professor Tiribelli, who I contacted almost 1 year ago, and he was very, available to collaborate with my young research group. I will start with the outlines of my talk. I will make a short overview about the notch family, then I will go deeply on my latest research topic, which is the role of notch3 in HCC. I will talk a little bit about the role of notch3 in angiogenesis and vasculogenesis, and then I will conclude with a few personal preliminary data. Notch family was first described in 1915 by a scientist named Morgan while he was studying the fruit fly, in particular the fruit flies with notches in their wings. And this is a very conserved pathway during the evolution, but also across the different species. And notch signaling is involved in very basic cellular processes, for example, cell proliferation, cell differentiation, apoptosis. And it has a very key role during the embryo development, in particular in organ formation, in the acquiring of different tissue functions, and also in the tissue repair. So since notch signaling is so involved in very basic cellular processes, of course, its deregulation can cause different diseases, and among these diseases, different type of cancers. So the family comprehends 4 receptors and 5 different ligands. Here in this picture, you can see the 4 receptors. They are named from 1 to 4. And they basically share the same structure. So they are membrane proteins with an extracellular domain and an intracellular domain. The extracellular domain contains the binding site for the ligands and some EGF-like repeats in a different number among the receptors, then a regulatory region. And the intracellular domain is very important because basically it owns the biological activity of notch receptors. The ligands are 5, and they are 2 classes of molecules, the jagged molecules and the delta-like ligands, jagged 1 and jagged 2, or JAG1 and JAG2, and DLL1, 3, and 4. They have common features, common functions, and but also their own unique functions. For example, DLL1 is involved in cell differentiation and in cell-to-cell communication. DLL3 is involved in cell proliferation and in the regulation of apoptosis. DLL4 is involved in neoangiogenesis, so basically in initiating the transcription of VEGF, and also in metastasis in tumors. JAG1, and keep this in your mind, is very important in vasculogenesis and angiogenesis, and JAG2 is involved in cell survival and cell proliferation. Here you can see a picture that describes the Notch signaling pathway. So as you see, both ligands and receptors are membrane molecules, and the ligand is expressed on the membrane of the sender cells, while the receptors are expressed on the membrane of the receiver cells. The receptors are translated in a unique protein that goes through 3 different cleavages. The first one in the Golgi apparatus, and so that the mature receptor is formed, and it goes to the cell membrane, okay? And then when it binds the ligand, there is another cleavage, which is operated by an enzyme, a metalloprotease called ADAM10, or sometimes by ADAM17, or more rarely by YW21, which is not reported in the slide. After this cleavage, there is a third cleavage, the S3 cleavage, operated by the gamma secretase. And at this point, the intracellular domain is free to move inside the cell. It goes to the nucleus, where it binds other proteins to assemble a big transcription complex that can initiate the transcription of different target genes. So the regulation of Notch signaling is involved in pretty much all the so-called hallmarks of cancer, as you see from this picture that I got from a review article. And it is quite surprising that for a small group of human tumors, it has a tumor suppressor activity, while for the majority of human tumors, it acts as a tumor promoter. So going deeply in my research topic, which is Notch3 expression, Notch3 role in HCC, of course, the first studies assessed the expression of Notch3 in HCC samples compared to normal tissues and cirrhotic tissues. So the first paper you see in the slide is from a group of the University of Bologna that basically has probably the largest scientific production about Notch3 and HCC. They performed an immunostaining of Notch3 in HCC samples and observed that a Notch3 positive staining only in the HCC samples compared to the normal liver, in which the staining was negative or very weak, and they obtained the same results for the cirrhotic tissues. They also reported that in the Notch3 HCC positive cells in which the staining was localized in the nucleus, there were higher levels of S6, which is one of the most important target gene of Notch, and is its transcription factor as well. These results were confirmed by other authors, as you see at the bottom of the slide, and also at messenger RNA level. Here you see the results from other authors who reported an higher expression of Notch3, messenger RNA of Notch3 in liver samples of HCC compared to the cirrhotic tissues, and the levels of Notch3 correlated with the levels of S1, which is another target gene of Notch3 as well. Also, other studies report the expression of NOTCH3 in different pathological conditions, so in other liver diseases, for example, PBC, PSC, and alcohol liver diseases. And again, it was reported that NOTCH3 expression was high in the pathological conditions, and it was very weak in the normal livers. In particular, the staining was localized in bile ducts and in blood vessels. Also, other authors observed a correlation between NOTCH3 expression and the clinical features of HCC patients. In particular, high level of NOTCH3 correlated with the presence of metastases and venous invasion, and also with a shorter overall survival, as you can see from the two Kaplan-Meier curves that I got from two different papers. And this is quite in contrast with results reported by the group from the University of Bologna, because in all their studies, they never found a correlation between NOTCH3 and tumor grading, which actually other authors reported, like the ones of the paper you see below in the slide, in which these authors correlated NOTCH3 expression level with tumor grading and also with the levels of the alpha fetoprotein, which, as you know very well, is one of the most important marker for liver disease, in particular for an aggressive liver disease. So NOTCH3 role was also studied in drug resistance in HCC and also in other kind of cancers. In fact, this picture reports results from different studies. This is a review article on different kind of tumors. So this basically summarizes the effect of NOTCH3 in increasing the resistance to different drugs in different tumors. And regarding HCC, again, the group of the University of Bologna, where let's say that this is the first study on this topic, and they used the cellular models, the FG2, which is the most famous hepatoma line, and the SNU398, which is actually an HCC cell line. And they tested the response to doxorubicin in wild type and in NOTCH3 knockout cells. And they reported that the response to doxorubicin was correlated to the expression levels of NOTCH3. In fact, after knocking down NOTCH3, the effects of doxorubicin increased. And they also show that this effect was due to an accumulation of P53. So basically, they speculated that the effect of NOTCH3 knocking down increased the cytotoxicity of doxorubicin, increasing the apoptosis rate due to the accumulation of P53. Then they obtained similar results, switching, the model, and because they studied the response to sorafenib in non-skid mice transplanted with the HCC cells. And they again observed that after NOTCH3 depletion, the effect of sorafenib, that, as you know, was probably for many years the first line therapy option for advanced HCC, the response of sorafenib increased. So they observed that this could be due to the effect of NOTCH3 on a protein, which is P21, which contributes to angiogenesis. So basically, depleting NOTCH3, it was increased the anti-angiogenic effect of sorafenib. Also, they tested another drug, which is brevanib. Brevanib is an investigational drug, so one of these drugs that was never used in real life, so no one patient was treated with Privanib, but it is interesting because it shares common mechanisms with the currently used drugs because it's a FGF and VGF inhibitor. So here they tested Privanib on rats, a rat model of HCC. And again, they correlated the response to Privanib with the expression of Notch3. And silencing Notch3 in rats, they obtained an increased response to Privanib. Again, the Notch3 knockdown cells showed higher expression and accumulation, let's say, of p53, but also the authors added some original findings because they found that in the Notch3 knockdown cells, 3 different enzymes of Krebs cycle, the TCA cycle, decreased, were downregulated. And this is actually consistent with the accumulation of p53 and with an increased rate of apoptosis because when the cell gets ready to apoptosis, it needs to slow down the glucose metabolism. So also they performed kind of a double check. So they depleted p53 in the Notch3 knockdown cells, and they treated them with Privanib. And they observed that the cytotoxic effect of Privanib decreased. So this is to prove that Notch3 effects on resistance to Privanib was mediated by p53. So they actually explained the functional mechanism of Notch3 p53 axis, which is responsible for modulating the response to drugs in HCC. They performed the Notch3 knockout, and they saw that this had an effect on a protein called cyclin G1, which is a cyclin, of course. And cyclin G1 is very important because it regulates the activity of another protein, which is MDM2, that probably you know very well, which is a regulator of p53. Basically, MDM2 is able to regulate p53 levels because it is able to induce the degradation of p53. So MDM2 is active when it's phosphorylated at T216, and cyclin G1 is responsible for the transcription of a dephosphorylase that dephosphorylates MDM2. So basically, it's quite hard to understand. Cyclin G1 can activate MDM2 through dephosphorylation. Okay, so when cyclin G1 is downregulated, MDM2 is not active and p53 is accumulated. Okay, so they basically explained the mechanisms by which Notch3 can induce resistance to therapy, or of course when it's knocked down or knockout, it increased the response to therapy. Okay. So Notch3 has also an important role in maintaining the stemness phenotype in HCC. And we all know that cancer stem cells in HCC have for sure an important biological activity in cancer onset, but mostly in cancer progression. So they are thought to mediate the response to drugs and an increased presence of cancer stem-like cells is linked to a shorter overall survival, even if we still do not know the exact mechanisms by which they act. In the paper you see in the slide, this author studied a molecule, which is a chromatin modifier factor, the LSD1 or KDM1A. And this is a molecule of which is very important in regulating gene transcription, is a demethylase, an histonic demethylase. So it's one of these proteins that demethylating the histonic proteins can open the chromatin, so change the chromatin structure, and so makes easier the access to chromatin, to the promoter regions of different target genes, to transcription factors or transcription complexes. So they found that in HCC cancer stem-like cells, there were higher levels of LSD1. And these levels decreased during cell differentiation. So then they linked the levels of LDS1 to Notch3, because Notch3 is able to induce the transcription of SIRT1. SIRT1 is responsible for deacetylation of LDS1. So therefore, they found that when there were higher levels of Notch3 in the cancer stem-like cells associated to HCC, they found higher levels of LDS1. Also, this higher expression of Notch3 in cancer stem cells was induced by the cancer-associated fibroblasts. So basically, they found a crosstalk between two different kinds of cells, the cancer stem cells of HCC and the cancer-associated fibroblasts. Also, in an ex vivo analysis, they found that higher levels of LSD1 and Notch3 and a higher proportion of cancer-associated fibroblasts in HCC correlated with a good prognosis. So again, this is another study in which Notch3 was involved in stemness features. In this case, the author studied another molecule, which is a membrane molecule, the CD44, which is considered one of the major markers for stemness. Basically, they correlated the levels of CD44 to the levels of Notch3. There is a direct correlation, as you see from the graph in the slide. And knocking out CD44, they observed a downregulation of Notch3. So basically, they suggested that CD44 is able to regulate the stemness property of the properties of the cancer stem-like cells through Notch3. Actually, this paper shows an association between CD44 levels and Notch3 levels, but it's, in my opinion, it's just an association. They didn't explain how, which mechanisms, explain this link between Notch3 levels and the CD44. So then Notch3 was also correlated, as I already mentioned, with angiogenesis. And in my opinion, among all the hallmarks of cancer, the role of Notch3 in the angiogenesis is the most interesting one. In fact, in HCC is a cancer with a high vascular density, so the vascular network is very important in HCC. And in fact, the first line options of treatment for HCC patients is a combination of drugs that includes neoangiogenesis inhibitor, for example, bevacizumab. And it's quite surprising that this aspect is probably the less studied among all the hallmarks of cancer concerning the role of Notch3 in HCC. So we think that silencing Notch3 could be, a good way to block neoangiogenesis in HCC. And so the study of Notch3 in neoangiogenesis could pave the way for, maybe a new therapeutic option. So to understand the role of Notch3 in vasculogenesis and angiogenesis, I will spend a few words about CADASIL. CADASIL is a disease that is caused by a defective formation of blood vessels in the brain, and it can cause leukoencephalopathy, and the main symptoms are multiple strokes and vascular dementia in the young adults. So it's a severe disease, which is due to a loss of function of Notch3. This because this loss of function is due to different mutations on the entire coding sequence of Notch3. And so far, more than 200 mutations, they are basically SNPs, single nucleotide polymorphisms, have been described as associated to CADASIL. So why am I talking about CADASIL during an HCC seminar? It's just because I want you to understand that Notch3 role is very important in the formation of blood vessels. So there are different studies that described the role of Notch3 in angiogenesis and in vasculogenesis during the embryo development. And for example, the picture you see in the middle of the slide is taken by from a paper in which the authors used a mouse retina model to study neoangiogenesis correlated to Notch3, and they basically observed that when both the alleles of Notch3 were knocked out, the vascular network in the retina of the mice was defective. So as you see, this one, just a moment. This one is the normal retina, and this one is the retina after vascular density is lower. Basically, they found that Notch3 was able to regulate the angiopoietin 2, which we know that it's a very important factor for angiogenesis. Also, of course, there are other studies in human pathologies, in particular in the hemangioma in children, and that show that an aberrant expression of Notch3 in the pathological blood vessels. In this case, the endothelial cells, which are basically endothelial progenitor cells, in the hemangioma samples had higher expression of Notch3, while the corresponding normal human vascular endothelium was completely negative. There are also a few papers describing the role of Notch3 in neoangiogenesis in tumors. This one is a nice study in which the authors evaluated breast cancers, in particular triple negative breast cancer, which is an aggressive tumor. And they found an overexpression of both JAG1, the ligand, which you remember is the one involved in the regulation of angiogenesis, and Notch3. Also, both of them, so the expression of JAG1 and Notch3, correlated with the density of blood vessels, and these blood vessels were positive for CD34. CD34 is a marker for newly formed vessels. So basically, these vessels were result of neoangiogenesis. Also, they found through an ex vivo analysis that in VEGF positive tumors, both the expression of JAG1 and Notch3 correlated with a shorter disease-free survival and a shorter overall survival. So again, an important role of Notch3 in the tumor biology and tumor progression. So to conclude, I'll show you some personal data. They are pretty few because for two reasons. Because we started to study Notch3 in HCC only 1 year ago, and because we encountered many technical problems in the experiments, in particular in the establishment of a knockout cell line for Notch3. We are working on YW7, and we really have in our time, so maybe we are almost done. But of course, this is the first step for all the other experimental tasks that we planned. So first, we performed an immunohistochemistry analysis, thanks to a collaboration with the anatomopathology department of the University of Pisa with Dr. Pinuccia Fabiana, which of course it's just a confirmation of previous results. We analyzed different grade HCC. We tested 3 anti-Notch3 antibodies, and the results you see are from the one who worked better, even if not in an optimal way. Anyway, in G1 HCC, so in well-differentiated tumors, the staining for Notch3 was, I'd say, negative in the liver parenchyma, while it was localized mainly in the blood vessels. And these blood vessels, so the Notch3 positive blood vessels, were also CD34 positive. So this means that these blood vessels were new vessels. So we speculated that probably in an early stage of HCC, Notch3 is expressed only by the vascular endothelium. In G2 samples, we started to find groups of Notch3 positive cancer hepatocytes, and the frequency of these groups increased in the G3 poorly differentiated tumors, as you see from the third panel. Also, in this case, maybe you see it in the smaller panel, in a group of G3 tumors, we found that a few isolated Notch3 positive hepatocytes were present in the stromal portal areas. So this probably means that these tumoral hepatocytes positive for Notch3 moved towards the stromal areas where blood vessels, lymphatic vessels are present. So basically, they locally invaded the stromal areas, so which is, of course, as we all know, one of the most important steps in HCC or in general in cancer progression. So we then confirmed these results in real-time PCR, so assessing the levels of Notch3 messenger RNA in the same FFP HCC samples. And we could observe an increasing trend of Notch3 levels going from peritumoral tissues, which were basically cirrhotic tissues for all the cases, to G3, so poorly differentiated tissues. So these results are in contrast with what was reported by the colleagues from Bologna, because actually we did, at least so far, we did show a correlation between Notch3 expression, and the tumor grading. So then we were wondering how Notch3 expression is regulated at transcriptional level, also considering that recently a great interest has been placed on the epigenetic regulation of different genes which are involved in cancer onset and progression. In fact, in this slide, you see the results of a study on a different kind of model. This is a study on T-cell acute lymphoblastic leukemia, in which the authors correlated the higher and aberrant levels of Notch3 with the activity of an enzyme, which is a KDM6A, which is again a demethylase, an histonic demethylase. So one of these protein that is able to modify the histonic protein, in this case the histone 3, H3, opening the chromatin structure and facilitating the transcription of different genes. So we recently established a collaboration with Dr. Vinicio Carloni of the University of Florence, who studies KDM2A, which is a molecule of the same family of KDM6A, in order to understand if there is kind of an interplay between KDM2A and Notch3 in HCC. So here from the picture, you see the mechanism of action of KDM2A, which is common to the other demethylase, histonic demethylase. So basically, you see how after the lysine, in this case, lysine 36 of H3 is demethylated, and this opens the chromatin. So first of all, we assess the levels of KDM2A in our setting, so in our HCC samples. And here you see the results of the immunostaining. We obtained a staining of positive staining in tumor samples, increasing from well-differentiated tumors towards poorly differentiated tumors, and the staining of the cirrhotic tissue was weak, very weak compared to the staining we obtained for HCC samples, while the LT liver was either negative or weakly positive. So again, we confirmed these results by real-time PCR, and again, we saw an increasing trend of KDM2A expression going from peritumoral tissues to G3. So in order to assess if there is an interplay between KDM2A and Notch3 in HCC, we performed the KDM2A knockdown in HUH7 cells, and we observed that knocking down KDM2A, there was a downregulation of Notch3 at messenger RNA level and also at protein level. So these are the results of 5 different Western blot analyses. So what's our working hypothesis? I told you that our main idea is that Notch3 has an important role in HCC angiogenesis, okay? We all know that the angiogenesis is induced by hypoxia. The main factor that responds to hypoxic condition is IF1 alpha. IF1 alpha is able to induce neoangiogenesis through the transcription of different genes that are involved in neoangiogenesis, for example, VEGF and other pro-angiogenic factors. Previous results show that IF1 alpha is able to increase and regulate the transcription of KDM2A. So far we observed an association between KDM2A and Notch3, only an association so far. So we, of course, are planning to go further and, for example, perform chip analysis and data sequence to confirm this association at a functional level. But if we will be successful in confirming this association at the functional level, and if we will confirm that also in our model of HCC, if 1 alpha is able to induce the transcription of KDM2A, we will find the association between Notch3 and hypoxia and neoangiogenesis, okay? So basically, our idea is to confirm a Notch3 if 1 alpha axis, okay? Just to have a double confirmation, we performed other experiments using our HUH7 models, and we observed the expression of a possible target of Notch3, because we said that Notch3 is involved in the maintaining of the stemness phenotype. So we evaluated the levels of CD133 that probably you know very well is one of the marker of stemness. So we downregulated Notch3 in our cells. We performed a knockdown for Notch3, and we observed that knocking down Notch3, the levels of CD133 significantly decreased. So going back to the other experiments, this is the histogram I already showed you. We thought if we evaluate the levels of CD133 in the sets that we have knocked down for KDM2A, we could demonstrate in a kind of not in an indirect way, okay, that KDM2A is able to regulate Notch3. So, and as you see, and as we expected, downregulated KDM2A, we observed a downregulation of Notch3, and this corresponded to an increased level of CD133 as well. Okay. So so far we could just observe an association between Notch3 and KDM2A, and I really hope to have new results next time, because we have different projects that we planned, and as you see from these slides, many ideas, and maybe too many ideas and too little money. So we are also working on fundraising, and I really hope to have more personal results to show you next time. So thank you very much for the attention, and I also would like to thank all the collaborators of the Patologia Generale of the University of Pisa, and all the external collaborators. Thank you.